Hi, everyone, and welcome to the Experimenters podcast, where we talk with women in biology and biotech about their work, their purpose, and the stories behind the science. I'm Jenny, a high school student passionate about biology and about exploring the deeper meaning behind scientific work. This podcast was created for those who are curious not just about how biology works, but why it matters. In each episode, we speak with women in science who are reshaping what it means to be a scientist, sharing their research, their journey, and motivations that fuel them. And I couldn't think of a better guest to launch this series with than Dr. Eva Nogales. She's a leading structural biologist, professor at UC Berkeley, and a Howard Hughes Medical Institute investigator. Her lab studies life at a molecular level, zooming in on how cells divide and how genes are switched on and off. What makes her work so fascinating is how she visualizes, especially cryo-EM, to see how these biological processes in action. Dr. Nogales, thank you so much for being here, and I'm really excited to learn from you today.
Thank you, Jenny. I'm delighted to spend some time with you.
So, to start off, I'd love to hear a little bit more about how your journey in science began. So, what first drew you in the world of structural biology and biophysics? And was there like a particular moment that influenced or sparked your curiosity?
So me ending up doing structural biology was a little bit of a stroke of luck, if you want.
Yeah.
So I was trained as an undergraduate in physics, in solid state physics. But I wanted what defined what happened to the rest of my life is that I studied in my country, in Spain, in Madrid, but I wanted to go abroad and to open my horizons a little bit. And when I looked into what were the possibilities for me to do a PhD abroad that will pay for my studies, I encountered an opportunity. I also, with a little bit of luck, was able to meet with the person that would end up being my PhD supervisor at a conference during a summer course. And he was, like me, physicist that turned into biophysics. So the idea is to use methods that are very physical and quantitative in nature, but to study biological process. So it was a little bit of encountering possibilities and taking chances, doing something new that I ended up really feeling very passionate about. But it was not directed towards structural biology by any means. I got to know what the structural biology was by doing it, rather than the other way around.
Well, thank you so much for sharing. And that's such a powerful reminder that like possibilities and inspiration can really just come anywhere.
Exactly. Yeah.
So let's go deeper into how your perspective shaped your science. So how do you see your identity or personal experiences kind of shape the questions that you ask in the lab or in science in general?
Yeah. So I think, again, the fact that I was trained as a physicist made me think about problems in a particular way that probably is different for someone that is maybe more trained in biochemistry. I think physicists have a good mathematical training and training in things like optics, things of that sort that tend up to be very useful because I was able to enter a field at the time when it was still very much in development, where you really needed to understand very basic principles of the technique. And that may have scared others with less of a quantitative training. But for me, it was a way to use my skills to otherwise embark in studies that were very novel for me because I was working with, biological organic materials, very different from what I had been trained on. So I just think that it was basically the way I had been trained and the way my brain worked and the way I was able to deal with techniques that were state of the art that gave me a little bit of an advantage in the field of cryo-electron microscopy when it was at the time, of my PhD and my postdoc is still developing.
Well, I really appreciate how like you connect your personal experiences into your work. It really makes your work feel more human, as I've read a lot about it. And that brings me into the role of visualization in your work, I could say, where seeing is understanding. So what does it feel like when you first see like your first a new molecular structure? And like, can you take us into one of those moments as well?
Yeah, I mean, it's very exciting, it's very direct. I always like to say that we humans are visual animals, so we learn a lot through looking at things and seeing their shapes and how they move or how they click one with the other, how they interact. So I think seeing these molecules is always very exciting because at the end it tells you about the chemistry and the physics of how these molecular components, cellular components work at the molecular level. I have many cool moments where just discovering the shape of the motion was particularly exciting. But I'm gonna choose one.
Yeah.
In that I find particularly interesting because it was very, very challenging, probably the most challenging project that we dealt with in my lab. And it took us many years to progress. And it had to do with a very large protein complex that is required for transcription of genes by the RNA polymerase II. And this complex in particular is called TF2D. TF stands for transcription factor. D has to do with the order in which it was discovered, and 2 is because it works with the RNA pol 2, the polymerase that actually transcribe genes that encode for proteins. In any case, this is a complex that is enormous. The polymerase is large, but this complex is more than double the size of the polymerase. And what was very challenging is that it is very flexible. And this is something that we were not aware of, and that in the way we are interpreting our data made it very confusing, and things did not make any sense until my graduate student, Mike Cianfrocco, was able to realize through his data analysis that one of the lobes of this complex, which is in very broadly speaking, has 3 parts. One of the parts move across with a very dramatic motion that is very rare to see in a biological molecule. And that the state of where that particular lobe was actually had to do with how the complex engages, binds to DNA. And the role of this complex is to bind to DNA in very specific manner to detect the presence of a gene and then recruit the polymerase to the right place. So seeing how this nuisance of this motion that made the analysis so difficult ultimately had a biological meaning that had to do with how the complex works was amazing, And then, once we discovered that, and with the advances of the methodology, we were able to get to the nitty-gritty, the details of how the binding to the DNA occurs. But even that is, we still have lots of open questions. So very exciting for us to visualize something that took us a lot of time, that visualization told us about how it functioned, and that it opened many other questions, which is the way science works always.
Yeah.
Every time you make a discovery, you get 10 more questions to ask.
Well, that's incredible. And honestly, I think in the future, I definitely want to see that as well. It sounds amazing based on what you're telling me, and like that you're witnessing something that probably no one else has ever seen. So now let's talk about microtubules, and they've been a big part of your research as well. So your lab has made major progress in understanding the dynamic instability. Could you explain how that behavior contributes to key cellular processes like mitosis?
Okay, so just let me start by saying that microtubules are biological polymers that are made of a repeating element, that is the alpha-beta tubulin heterodimer. Let's call it tubulin. So tubulin self-assembles, making a tube, a structure that, because it's very small, we call microtubule. And there are hundreds of them in a regular cell, doing many things, from organizing the contents of the cell to actually segregating chromosomes during cell division. So This is an ancient protein that is very abundant in all our cells, absolutely essential, and actually a target of anti-cancer agents because of its involvement in cell division.
Yeah.
In any case, one amazing property of microtubules, and that was one of the reasons why I was so attracted to it as a physicist, is that it exists in a metastable state, meaning it uses energy which comes in the forms of GTP hydrolysis. So GTP and ATP are kind of energy coins, if you want, in the life of the cell that you can burn to, by hydrolyzing the last phosphate in order to generate a burst of energy that typically changes the conformation of proteins and allows them to do their function. So in the case of tubulin, the process of GTP hydrolysis, so it binds and it hydrolyzes its GTP. And when it hydrolyzes it during the process of self-assembly, it generates a microtubule lattice that is not stable. And that at a certain point, stochastically can depolymerize. I said stochastically, but also it can be regulated in the cells by proteins that has as a function to make it more or less likely that the microtubule will switch between a process in which it's growing to one in which suddenly it is assembled.
Yeah.
So this is dynamic instability, is this metastable behavior of microtubules in which they are able to switch between phases of growth and phases of shrinkage. And it has a structural basis that relates to how tubulin changes conformation inside the microtubule after GTP hydrolysis to make it more unstable. So this is something that we've studied for a long time, and we continue to study because all the regulation that occurs in the cell.
That's amazing how
But I don't know if I answered the question that you asked me.
Yeah.
Exactly, but yeah.
Yeah, I think it's like amazing how precise like cells have to be such an essential part of our life as well. So
there has to be many layers of regulation, in many cases with certain redundancy, with many signaling, so layers of information that the cell needs to be able to integrate, to respond to changes or to maintain what we call homeostasis, which is happiness at the cell level.
I think that's why I just love biology so much, because of like this tiny little features that are so essential to us. Staying on the theme of seeing structures, when you visualize something like the transcription machinery or PRC2 for the first time, what goes on through like your mind, like especially when you first saw it as well in the beginning of your career?
Every time we see a new structure is, how can we mine it? How can we look into the details and understand how it works? So there are certain things that we always do, like, for example, put the structure in the context of all biochemical and cellular data that it exists, as well as even genetic data that relates to diseases. So a very obvious thing to do is when we're studying a complex for which we know certain mutations give rise to certain diseases, that we map where they go and we understand how can it be that in those positions this could have an effect. In many cases, the structure of the protein complex by itself may not necessarily be very meaningful, but what it helps is that these proteins do a certain task. For example, you mentioned PRC2. This stands for polycomb repressive complex 2. This is a complex that carries out an enzymatic activity on chromatin that makes chromatin become silenced. So there are many layers of regulation because you don't want to silence part of the genes that are required for the life of the cell, or vice versa, be transcribing a gene that should not be being read for that type of cell. So there are many layers of regulation, and many of them have to do with pre-existing state of chromatin. So these complex have to interact with nucleosomes, which are this complex of histone proteins and DNA that wraps around them. And is very important. How does it interact with this nucleosome? How can it engage with it? How can it read if there is a nucleosome of a certain type or another? And all of that is what we can see when we look at the structure. Yes, can we make sense of all the biochemical data now in a physical structure where we can position each one of the amino acids? And can the structure now give us new hypotheses about how it works that we can go on testing both in the test tube and in the cell?
So it sounds like every time like you find something new, it opens the door to so many new
Always.
questions.
Yeah, that's correct. Yeah, that's correct.
So zooming out a little bit, what role has cryo-EM played in changing the field, and what does it answer in the field of molecular biology?
Yeah, so cryo-EM, cryo-electron microscopy is a structural biology technique.
Mhm.
Structural biology is the field of molecular biophysics that is dedicated to obtaining the structure of biological components, and for a protein, what is the path of the polypeptide chain that ultimately gives it a certain shape and certain chemical and physical properties. So the ruling technique for many years, and still very, very important, is X-ray crystallography. It's a technique that uses powerful X-ray sources and crystal forms of your protein or your protein complex of interest, which are not easy to obtain, to then get diffraction data that through computational analysis can be utilized to generate what we call a density map, something that defines the shape of the protein. That technique, very powerful, has certain limitations. Certain proteins are not easily crystallizable for many reasons. One, because you cannot obtain it in large enough amounts to get the concentrations that you need for crystallization and to do many, many trials and the different crystallization conditions. Because they are flexible, and then when they are flexible, it's very hard to trap them inside a crystal in a fixed position. There could be many reasons. So cryo-electron microscopy is a technique that does not require crystallization, and that is more direct in the sense that things are actually truly imaged rather than getting a diffraction pattern that then you have to analyze in different ways. So it has challenges of its own, but it's actually ideal to study things that are very large and hard to produce in large amounts and have to put into a crystal that tend to have movable parts, and cryo-EM can deal with that and actually describe those movements. And you can study things that you can obtain in very little amounts, like, integral membrane proteins or endogenously purified cellular components. So it has opened the door to make structural biology more generally applicable to any biological macromolecule. I think that is the value that it has had, and with time is getting to be to allow higher resolution. This is why something that used to be just an academic technique now has become very broadly utilized even in biotech and pharma, where is used to visualize small molecules that are therapeutic, possible therapeutics as they bind to their targets. So a technique that has really generalized the use of a structural biology to describe the structure and the conformational states, the different types of motions that macromolecules can undergo.
I like how you say that it goes beyond academia and it could be generally applicable. And I think that's like the main purpose of this podcast, is to see how we can apply these studies into our real lives.
Yeah, so one obvious, so I'm a scientist that is guided by curiosity. I just want to understand how the world works. But the type of information that I generate in my studies can then be utilized by people with more practical pursuits, like the identifying or improving therapeutics. Many therapeutics are small molecules that act by binding to the surfaces of proteins and altering, normally disrupting, the functioning of that target protein. And therefore, visualization of the protein, to give you an idea of what are druggable sites, or visualizing the drug bound to the protein and seeing how, understanding how it works, understanding how it can be improved, how it can be made to be more specific or binding with higher affinity or something like that, is something that is very essential in the process of drug development, and that now has incorporated cryo-EM, and as it has become a high-resolution structural biology technique.
Thank you so much for sharing. So now thinking about the bigger picture, when you're working with something as fundamental as gene expression, do you ever reflect on like the ethical or societal implications of what you're helping to uncover?
Yeah, so the kind of studies that we do, they don't have an obvious way in which can be utilized for bad, although you never know. So it is not like studies like my colleague Jennifer Doudna is carrying out with CRISPR, where as soon as she realized that the CRISPR systems could be used for genome editing, it was very obvious that this was, a tool that could be used for changing germline or things that could have more ethical implications. I think in general, generating knowledge is a positive thing. Understanding our world is a positive thing. In the biomedical arena, we know that the work that we do is very important to understand human disease and to improve it. And I tend to think that the work that we do really has many positive connotations and not that many negative. But maybe my brain is not so twisted that I can think of how that kind of information could be used for bad. I think, maybe this is a more general question, but I play a role as teacher, both of members of my lab, but also I teach undergraduate and graduate courses at Berkeley. And I just think more of the moral implications that it has to be a teacher, to be someone that is contributing to forming individuals that hopefully will grow up to be critical thinkers, that know how to gather information, that know how to synthesize it, that know where to go, to have sources of information that are reliable, that are able to, to put this information together, work on the data and come out with their own conclusions, and that they know how to engage in discourse with others that may or not think like them, to actually learn and progress as a, maybe as a scientist, but maybe just as a citizen. I think that is a part of my job as teacher that I think is very important, and that I take very seriously, because it does have moral consequences of, how I'm influenced, hopefully in a positive way, the way people think, gather information and make decisions that are informed.
Yeah. It's so refreshing to meet educators that just love what they do and making a positive impact on their students. And like your work just touches like so many layers beyond that as well. So let's now.
Just very quickly, because I just want to see a movie that is called The Last Class. I don't know if you know it. It's like a document. It's a documentary on the last semester here at Berkeley of a now retired professor, Robert Reich, who is a very important figure. He's been a very important figure in politics and economics in this country. And that was an excellent professor that really in the movie, it comes across as someone that was very dedicated to this role as an educator. And I was talking to you and I was thinking of that movie. I highly recommend it.
Oh, that's
The
amazing.
Last Class.
I'll make sure to check it out. It sounds amazing. So let's shift to legacy and impact. So what do you hope that your lab will be remembered for and scientifically and both in the community?
Well, I think I would like to think that my lab has been very brave in engaging with areas of research that were considered very difficult and that many didn't dare to trample on, and really open the door by demonstrating feasibility and getting initial results. So and to be a little bit, I would say, leaders to a certain degree in certain fields of structural biology study that then have encouraged further work from others, which is what is important. Whatever your work do, whatever you do as a scientist, you do it building on what others have done. And if your work is of quality, others will be able to build on it. And I think this is something that I feel very proud of, that both on the microtubule tubulin field and in the field of gene expression regulation, my lab has done critical work at certain states in the history of that knowledge that were critical for others to really become engaged with that field and then take it forward.
Well, thank you for sharing. And finally, for like a message to the next generation to close off, if you could tell a young woman who is curious about science one thing, and not just like what science is, but why it's worth doing, like what would you say?
Well, I'm someone that feels really privileged to be doing what I'm doing. That's why I think maybe I will never retire. I think science, especially in an academic environment. I'm double privileged because I do this work at UC Berkeley, which is a fantastic place for many reasons. This allows you to work all your life surrounded by young, talented people. So people come through my lab, they typically spend between 2 and 6 years, either as undergraduates doing research, as graduate students, or as postdocs that are training to then start their own labs or go to biotech companies and things like that. And every time someone comes, it brings new energy, new enthusiasm, and they come and go, and sometimes it's sad to let them go, but many of them are scientists that I get to interact with. And on the other hand, I'm always surrounded by young people with that kind of energy. I think that's very unique to doing science, and that there is a general interest and understanding among scientists that we are all in here following the rules of the scientific method, pursuing the truth, sharing information, able to take criticism to improve our own work and improve from it. And that kind of a spirit is really wonderful. It's a world very international where people come from all over the world with that drive and passion to do research, and it really is very beautiful in that sense. And you were referring to that moment when you learn something, know it for the first time before you transmit it to the rest of the world, that eureka moment. They're really wonderful. So there is a lot of hard work, a lot of apparent failures from which we learn and eventually hopefully get to a happier conclusion. And when, after all that effort, when it comes to fruition, it's just very, very rewarding. And we know that we're pushing the frontiers of human knowledge, and that is just so rewarding.
Yeah, that's such a meaningful message to end on. And thank you for encouraging the next generation with heart and honesty. Dr. Nogales.
Thank you, Jenny, for what you're doing. This is a beautiful project, and I hope that it helps others, and that you should be feeling very proud of having thought of this and doing it.
Well,
Thank you
so much for being here. and opening up about your work and what drives it. And it's been amazing to hear on how you combine your discovery, creativity, and purpose, and how much of an impact that you've had by just simply looking deeper into this field. To everyone listening, thank you so much for joining us on our first episode of the Experimenters podcast. We'll be back soon with more conversations that explore the why behind biology and spotlight the women who are transforming science from the inside and out. Thank you so much again, Dr. Nogales, for joining me, and thank you for listening.